There is a financial cost of having to go through research for this. It is apparently, another hurdle. So they were looking at whether or not
I don't think this is switched on.
It's not.
I don't think so. As in, this is on, but I don't think it's linked to anything.
Is it on?
Yeah, it's on, but I don't think it's working.
No. Oh, there's another button here to turn this on too.
That's it.
All right. Is the live stream running?
Everything's good to go.
Okay. There'll be a couple more people straggling in, but I thought I would start the introductions of this second GSI Technology Seminar Series of the spring. This is kind of a slightly different flavor to our usual GSI Seminar Series that have academic speakers, but today we're having a biotech industry speaker with Dr. Michael Ko from EpiCypher. Before I introduce Dr. Ko, I first wanted to make an announcement about the Boston University Bioinformatics Master Summer Internship Program. This is one of our educational partnerships between the GSI and the BU Bioinformatics Program. Dr. Adam Labradorf is in the Department of Neurology as an assistant professor, and he also is the director of the master's program for bioinformatics students. And he's run this program pretty successfully in the past, where he helps connect master's students in the bioinformatics program with PIs that might have a summer project that they would like a student to help out with and get some mentoring from the bioinformatics program as well. There's a website that's now up on the GSI that will advertise further on other emailings. We're looking for applications due March 1st. If you have a research project for the summer that you think a master's student might be interested in, please submit those by March 1st to the GSI email. We'll have other announcements by email on this, so keep an eye out for that. It is my pleasure to be introducing today Dr. Michael Ko. Michael Christopher Ko, or you just go by
Michael.
Michael, okay. He is the Chief Scientific Officer from the company EpiCypher, which develops these kits and these techniques for profiling chromatin, nucleosome modifications, as well as transcription factors for an assay also known as cut and run. We invited Dr. Ko to present their company's technology because we have this GSI pilot grant workshop competition. Those applications are due April 7th. And we'd like to center those applications around some seed funding that the GSI could provide for BU labs within the GSI to try out these technologies. And we have two themes this year. We have cut and run that Dr. Ko will present, and in the previous earlier seminar it was about split seed. So we're kind of trying out these new genomics technologies out and seeing what can advance your genomics research. And for other faculty that are interested in hosting GSI seminars, we are still open to faculty and even graduate students and postdocs who are interested in hosting GSI seminar speakers. If you're interested, please reach out to us. Dr. Michael Ko is the CSO of EpiCypher. He was actually, he's been there for like 8 years with EpiCypher, 7 years. And prior to that, he was actually a PI at Albert Einstein with his own lab working on DNA profiling and chromatin profiling in cancer. And before that, he was a postdoc with Stephen Buratowski, who was also a well-known transcriptional regulation PI studying in yeast. So he comes from both the biotech background and the academic background. And so after the technology presentation here, there'll be a lunch in the back for those of you who signed up to have lunch with Dr. Ko. He can talk more about the technology as well as his perspective of bridging the two different arenas of research, both in academics and in industry. Without much further ado, Michael Ko. Thank you.
Sorry, I was knocked off your little mic there. Oh. Oops. Thank you, Nelson, and also thank you to Abigail for helping to put this together. I'm going to give a talk about our technology, but I always prefer to do it by showing what we did with it, rather than just simply saying, here's tech A, here's tech B. And so in that regard, we are extremely collaborative. We get pretty much all of our funding for R&D through the NIH. And so that with that in mind, we basically always want to publish. We're highly engaged with the community with regards to developing new tech and showing what we did with the old stuff. So I'm going to show you some toys, but then also what we did with it. And if we have enough time, I'll show you some stuff in R&D. And I'm not sure if we're going to have enough time because I got way too many slides here. And so just to take home message, since we are an epigenetics and technology development company, one thing I would note that we've learned when we started, and well, we had this idea when we started, and we've learned that we were correct in this assumption. And this is that if you want to work on chromatin research epigenetics, you really need to work with histones and nucleosomes. Working with peptides as a surrogate, not so helpful. And I'll give you multiple examples of how this has sort of been shown to be the case and how we've been led in some very strange directions. I'll show you, of course, that, well, data-wise, and of course, Nelson had asked me to comment on sort of what we've learned. I'll show you that most antibodies that are used for histone, to detect histone PTMs are not fit for purpose. All of this has been published and will continue to be published. There's no secrecy in this regard that we're holding secret data internally. And then I will show you, in fact, you don't even need reader domains. If you want to do PTM mapping, you can actually use, reader domains themselves. And in fact, you can do some extremely interesting things with them. But all of this requires spike-ins, because if you have the target as a spike-in the experiment, you can actually tell if it's working effectively, rather than simply looking and yielding data and then trying to reconcile it with what you believed was going on. Okay, and like I said at the end, if I've got enough time, I'll talk about our DNA methylation mapping, multi-omics, and then also an assay that we're developing with NEB for open chromatin mapping. Okay, so how do I actually do this way? So, like I said, peptides are not little nukes, and so the reason is because of the way things are presented. So you've all have seen this sort of original idea of the nucleus containing the chromatin wrapped inside it, being a packaging, but also a signaling material. The original Luber-Richmond structure from years ago, beautifully elegant structure. When we saw this, and when it was originally published, the majority of the histones were classed as structural. About 70% of the histone map, or histone sort of mass, is easily identifiable in the structure. And it's inside, it's involved in multiple points of histone-histone and histone-DNA contact. About 30% of the mass, primarily the tails, was not visible on the structure. And so for that very reason, to give a vague idea of the relative mass, it was designed as, it was shown and depicted as sort of protruding. And if you look at any subsequent studies, in fact, when I was a postdoc in Steve's lab and then also my own lab, when we were identifying new PTMs, we were, of course, showing how they interacted with each other and how they were presented in relationship to each other on the tails. And of course, we always presented the tails as sticking out, because that way we've got more space to actually put things on top of it. And it's a very short distance between depicting it this way and then believing this is the way it is. And the problem, of course, is that it isn't. The tails are not easily accessible. Despite what I probably said in various talks down the years, they are not easily available for recognition and modification. They're actually in contact, in very close contact with the nucleosome surface, in contact with each other, in contact with the DNA. Anything that wants to engage these tails for recognition or modification has to engage with this surface. It has to. This is a transaction that has to occur. It is obligate, it is not an optional extra. So therefore, peptides representing the tails are not exactly what anything sees in vivo. And so if we remember the players that we're talking about here, there are writers that add modifications, readers that see them, and the erasers that remove them, and then of course there are remodelers on top of this that move chromosomes around, move nucleosomes around, give access to regions of open chromatin. And then this is of more than sort of blue skies academic interest. There is massive engagement in drug development. So I started working on chromatin just soon after the original Dave Alice paper was published on GCN5. At the time I was a postdoc in Steve's lab working on gene expression. All naked DNA. The second I saw this GCN5 being acetyltransferase, and that was known to be a transcriptional regulator, dropped it all. And since I even started that, that's some years ago, it's embarrassing, in the mid-90s in my postdoc, we are now well within drug development. These things are already in patients. They're past phase 1, 2, 3. There are some of these drugs for various PTM, for various histone or epigenetic modifier targets are already inside patients and being used to treat disease. Okay. So within EpiCypher, we had this idea that peptides weren't going to cut it, so of course we had to learn how to make nucleosomes. So I want to call attention to this. You can start making a single nucleosome relatively straightforward. You just need 8 histone proteins, well actually 4 because they form an octamer. And so you have the 4 histone proteins wrapped by 147 base pairs of DNA. This is a mononucleosome. You can biotinylated and then immobilize it onto beads. You can then, once you can do this at scale, you can then start to do interesting things. You can put in variants, you can put in ONCA mutations, you can make remodeling substrates, you can barcode it. But the centerpiece of our portfolio are the designer nucleosomes, and these contain the post-translational modifications themselves. They are not surrogates, they're not analogs, they're not enzymatics, they are semi-synthetic and pure. And we can now basically make any PTM that we've gone after. Currently, we've got a focus on ADP ribosylation, for example, as one of the last ones we've decided to address. We can make any one at any position, and we can also do them now, most recently, also we can make heterotypics, where we can do different modifications on different histone tails. Okay, so we've been supported, extensively supported in all of this through the NIH SBIR program. And so they've given us very generous amounts of grant money down the years to basically, and we look at them as being in 3 stages of funding. And the first one is we get grants to develop a new type of PTM. So for grants, for example, learn how to make acetylations at scale, learn how to make ubiquitolations, etc. Then we get another series of grants from different agencies, for example, from NHGRI, to develop genomic assays with these new technologies. And then finally, once we've shown that we have the assays in place and we have the ability to make new PTM classes to fit into the assays, etc., we then start getting money from different agencies that are interested in specific disease types. So in other words, to interrogate that type of disease. So the NIH through the SBIR program, like I said, has been extremely generous. And we've, I think, currently got about 12 or 14 active grants, all sort of stages of doing this. So to show you what we did, the first technology, the one in fact that we bet the company on all those years ago, was the attempt to standardize chip. And it was to actually put PTMs of chip is pretty much a black box. It's been used by genomics, or people who are doing genomics for years, but it's notoriously difficult to do. And it's also notoriously long, it's not proven, it's not good for HTS, it's expensive. You don't even know if it worked until you get to the end, etc. Everybody claims that this antibody works and that one doesn't, and everybody's basically saying that based on yielding data. Signal over noise is terrible, horrible. But we said, okay, we can fix this, at least we thought naively we could. And the way that we were going to do it was by making PTM-marked nucleosomes and throwing them straight into an experiment. In other words, you were going after H3K4 trimethyl. You wanted to know it works, so just throw in a spike in nucleosome and then monitor the recovery. But you don't just want to monitor recovery, you also want to monitor the specificity. So we said we would make panels. So in fact, this is what we bet the company on, that we could make this panel and people would want to buy it, and so we'd all be rich and we could all retire. So we made the MetStat panel, which was the first one, and subsequent panels, by the way, have been similarly comprehensive. But we've got here H3K4, 9, 27, 36, H4K20, mono, di, trimethyl, and unmodified. That's 16 different nucleosomes. It's actually 32 because we've got an A and a B, different barcodes of each. Make them individually, barcode them, pool them, spike them into an experiment, and then simply ask, I've got an antibody. I want to know if it's going after a various target. Therefore, for the antibody to pass, it has to show less than 20% cross-reactivity and greater than 5% recovery. These are not complicated metrics. You would feel that they are reasonable. The only problem is that despite them being reasonable, they're failed so often by commercial antibodies. Here's a block of 263 unique catalog numbers to lysine methyl, 129 to acyl, so there's about 400 antibodies in here. Commercial catalog numbers doesn't even take account of lot variability. Failure rate inside that data set is 71%. So in other words, we spent a lot of money just basically putting stuff in the freezer that was of no conceivable use. So we asked, okay, so if somebody enters the market, the chances of them finding an antibody to monitor the target they're interested in is less than 3 in 10. Nobody basically randomly buys antibody. Everybody buys what their colleagues have been using. So we went and we asked, well, okay, how about the top most cited antibodies? We had top 10 most cited targets, top 3 most cited antibodies per target, that is 30. We had 24 of them in the data set. The failure rate was 80%. It was actually worse. These antibodies, interestingly, have largely been selected to be very, very good at the recovery metric. They yield data. They consistently yield data. Sadly, it is not the right data. And so the problem I would note is that the field is littered with these. In fact, a lot of the big gorillas working on PTM mapping, like ENCODE, sadly chose mostly the wrong reagents because they were going after the ones that were based on recovery. We also figured out some years ago, so we've published all this, at least in the first instance. We also, of course, asked, well, okay, it's screwing up the recovery of the PTM standards, but is that actually what's going on inside the cell? So here's a block of data from the original study back in Molecular Cell back in 2018. Here we're looking at 3 antibodies to H3K4 trimethyl, and went inside, we're normalizing to the on target in red inside the red box, so that's 100% on target. And what you see here are 3 antibodies. 2 of them are showing profound cross-reactivity with dimethyl, 60 to 80%. It's worse than it actually looks because these, in this case, the spike ends are equimolar, but inside cells, the PTM targets are not. K4 trimethyl is actually very, very rare. K4 dimethyl, much more abundant. So that's 60 to 80% cross-reactivity with a target that's 3 to 5 times more abundant. And when you look at the ChIP-seq data tracks, you can actually see this. Good antibodies to K4 mono, good antibodies to K4 di, and then 4 antibodies to tri. These ones are specific. These 2 are the ones that are cross-reactive. Almost all of the literature on K4 di, or sorry, tri, is actually K4 di, which is a profound problem with regards to interpreting the biology of what this marker is actually doing. Also, if you're doing things looking at demethylases, for example, which convert the tri to the di state, you're now in deep, deep trouble. Okay, we figured out what was the problem. That is ChIP. That is not the same as what we've all been using as the gold standard approach, which is a peptide array. Biophysically, they are very, very different. And in fact, when you compare the data sets, you look at the specificity metrics that are given for various antibodies in the 2 settings, and the Pearson correlation was about 0.23. You were worse than the coin toss. You yield lots and lots of data doing this, but sadly, it is not predicting what's going to actually happen inside the ChIP experiment. Okay. So then we, of course, had been falling out of ChIP for all the reasons I mentioned earlier, older tech, and we've started looking at this newer one that had been sort of worked on by Uli Lemley and then subsequently by Steve Henikoff. And this is an immunotethering approach, which internally is known by the name Katana, but it's basically cut and run, cut and tag, and then there's sister technologies that we continue to develop. And so the approach here that the setup takes is different. Here you take the cell and you immobilize it onto a solid support. So rather than in a ChIP experiment making a mess and then trying to fish it out, here you take a cell and immobilize it onto a solid support, being a magnetic bead, and you permeabilize it. Then you take an antibody and you basically add it to this mix, and it diffuses into the cell and finds its target, be it a PTM or a transcription factor. So it finds its target in situ, and then you add in the case of cut and run, protein AG MNAs, and the protein AG finds the antibody via the constant domain. You activate the MNAs, it cleaves wherever it is adjacent to. It releases its target, which now diffuses out of the cell into the supernatant. And now here's the killer step that actually makes this all work. You pull down everything else being the background on the solid support. By doing this, you have completely shifted the signal noise in your favor. ChIP is a signal noise problem. It's always been a signal noise problem. By doing this step, you actually shift more towards the signal and away from the noise. This does change everything. It reduces the cell needs. So 500,000 is the standard, but we've been doing a major drive on 10,000 cells for a large the Immunological Genome Project. You do actually need a lot less sequencing depth. So standard ChIP between 30 and 100 million reads, depending on your target. Here, 3 to 5 million mappable reads is all we go after. So generally, it's 5 to 8 million total reads that we're going after in any given experiment. We have developed robust protocols for sample prep and transfer. All of these are made available online and through our kits, etc. There's no secrets. And the reason, of course, we did this is because we're now transferring material within our services group from locations to us. And this would include people working on drug development, and I'll call attention to some of that. We've developed a protocol for native and fixed cells depending on your target. We've shown that it's suitable for a broad range of targets. I'm going to largely speak about PTMs, and I'm going to show you some data in passing on transcription factors, readers, and ATPases. And then the most interesting thing for us, especially with regards to the capability of this technology for drug developers, we have fully automated it. We're now capable of doing up to 4 plates a week, 96 well plates on our robotic systems, all the way from cells to libraries. This is thousands of data sets in the last few months, all basically used on genomic data mapping. Okay, so in addition to actually having access to the technology at the beginning, that meant we could create standards. In the case of ChIP, people were resistant to take the standards on board because they already knew how to do ChIP. They didn't need them or thought they didn't. In the case of cut and run, since we had the tech from the beginning, we can standardize it immediately from the beginning. So we do exactly the same thing. We have our 147 base pair with an embedded barcode, and we have 5' and 3' linkers. So this position nucleosome is immobilized onto a biotinylated bead. And then, of course, we make barcoded panels just as before. And here we have K-Metstat again, and we have K4 mono, di, tri, and unmodified, K9, K27, 36, exactly the same panel as before. The reason for this footprint, by the way, is because it is now compatible with cut and run, which I've mentioned, but also cut and tag, which we're not going to talk about, but I can follow up on if anybody wants to talk about. These spike-ins are on magnetic beads, and you can probably see where this is going. They're spiked into an experiment with the cells also on magnetic beads. Now you add in the antibody, it goes to its target, be it inside the cell or out. You activate, and then you check what's basically sitting in the supernode. And when you do this, and you basically go and look at antibodies, whoops.
Battery cut out.
It's fine, I can speak up, it's no big deal.
Here, let me see what's the thing. I'll get a new battery. Here it goes.
It's still red though.
Is it?
Yeah. That's okay, I can speak up.
Okay.
Okay, so I'll speak a little louder. So the antibodies again are checked with regards to their capability. And what I call attention to is the fact, again, we have K4, mono, di, tri, etc., the whole way down through metstat. And you can see as we walk down through the panel, there's an antibody being identified to every single one of the targets in the panel, with the exception at the time of H4K20 monomethyl. These antibodies have always existed. It's just been a case of finding them to use them in the appropriate experiments. Okay, in addition to us going after the specificity, we're of course also going after efficiency. So I mentioned that a standard cut and run experiment is 500,000 cells, but sometimes you don't have that amount of material. You're working with very precious primary sorted cells, etc. So we of course have been interested in developing new reagents that can go further. This, by the way, the assumption that every reagent that will work as you drop it down, it's dependent on antibody efficiency, which is a metric of on-off rates, avidity, etc., etc. So here, for example, we've got a new antibody that we've been developing internally to K36 dimethyl, and what you can see is the peak structure at 500,000 cells is held all the way down to 5,000. These antibodies are a very rare breed. So it's about, like I said, about 75 to 80% of antibodies don't have the metric in that were shown to be good enough for us to actually do the PTM mapping. What I can say is less than 20% of them, of the good ones, have the capability to go all the way down here. All right, once you've got these reagents, you can do some amazing things. Just looking at the maps is an indication of the amount of information that remains to be discovered. So for those of you who are trainees and we talk about K4 tri or K36 tri, etc., there is so much we do not know, and we've been taking for granted all these years, because the PTM maps that we're generating now are an amazing resolution compared to anything that came before. It's easy to see here, for example, the distinction between K4 mono and K4 tri is easy to understand. Everybody mentions K4 tri is a mark of active promoters, mono is a mark of enhancers. But what we've done is massively underestimated the relationship between other methyl states at other positions. This is K36, for example. In yeast, the mono-di-tri states generally overlap each other because they're all catalyzed by the same enzyme. But in metazones, there are distinct enzymes that actually stop at different states. So here, for example, you can see that K36 tri is found inside gene bodies, whereas K36 di is not overlapping it. It's actually found in intergenic regions. K27 is fascinating. All PRC complex mediated, the same enzyme catalyzes the mono, di, and trimethyl states. Everybody talks about trimethyl, but look, mono, di, and tri are completely distinct states. The complex is being controlled to put the PTM of choice at different positions, and it is not doing it to recruit antibodies. It's obviously doing it for various forms of biology. We also have access to the entire genome, as far as we can tell. You might imagine that because it's a three-dimensional assay, essentially you're looking at nucleosomes and chromatin in the native context. Well, if the region is compressed, can you actually gain access to it? Recently, the human genome T2T build, the telomere to telomere consortium data was released last April. This gives access to the last 8% highly repetitive regions of the genome. It's an amazing release. I think any of you who are doing genomics should really take it on board. But here we're actually looking at CentBay. This is a histone H3 variant that marks centromeres, specifically the core centromere. You can see that inside this location, we can also map into it K9 di, K9 tri, and K27 di, not any of the other states that we're going after. So in other words, we are gaining access to the regions of the genome that would always have been assumed to be inaccessible. They're defined as such, in fact. Oops, oh, we're back. Thank you. Spare my throat. So regions that are the original definition, in fact, of heterochromatin versus euchromatin was apparent high density and is presumed to be inaccessibility. And yet to the actual experimental setup of cut and run, and presumably also the cut and tag, these regions are available for yield for data generation. We don't just work with PTMs, also works with transcription factors. Here, for example, we're looking at TF-FOXA. We're simply just doing the experiment, enriching for TF, looking at the regions that are enriched by it, confirming that here's the motif that's supposed to be bound by FOXA1. So if we discover that 30% of the motifs in this data set are actually enriched, and that makes sense, you're never going to get 100% for TF because you also get formation of other higher order complexes. It's targeted not always by its direct DNA binding. Here's an example of the peak structure. The one beautiful thing is we can make standards now for anything. So here, once we've got an antibody that works, in the case of PTMs, you have to make the standard and find the antibodies. In the case of TFs, you've got to kind of go the other way around. We find the antibodies that work based on all the available data, and then we make a standard to accompany that antibody. Essentially, we know that this antibody to FOXA1 was made by this immunizing peptide of 50 to 60 residues. We simply couple that onto a nucleosome, and then we use that to follow through all subsequent experiments. The ability of a spike-in is immense. You do the experiment, you look at the data, you look at the standards. Did the standards work or not? If they didn't, you stop analyzing. Why are you continuing? You just discard the experiment and go back again. So this is now the approach that we're taking to as many different targets as we're going after. One of the home runs that was done with the technology years ago was the ability to go after ATPases. These are the remodeling enzymes. The chip data sets on these are god-awful. And the reason is because of the way that ATPases actually engage with chromatin. They touch down, they burn ATP, and they leave. They do not dwell on chromatin. But there is one way of trapping them in situ. It's the burn ATP step. It was shown years ago that if you actually dropped ATP levels, you could increase the dwell time and increase the frap, the frap time of actually ATPases. And remember, when you permeabilize cells to do a cut and run experiment, the ATP levels plummet. And so what we immediately discovered on the first pass is every single ATPase we managed to capture it in situ, all the remodeling ATPases. Here you're just showing enrichment, but when you actually look, you can actually see the beautiful peak structures. And this is, I can just comment, this is way cleaner than anything that had ever been seen before in cut and run. In a chip experiment to do an ATPase, you normally needed more than single formaldehyde. You needed multiple different cross-linking agents to be added at the same time. Very heroic, hideous backgrounds. Okay, so how sensitive is it? And so at the time we said, okay, are we simply able to detect when something is there or isn't? If we're going to use it for genomics, we're also going to use it in clinicals, we're also going to use it in drug development. We want to know how sensitive it is. So we said, can it detect a quintile change? In other words, a 20% change in occupancy in response to a stimulus or a change. And so here we've taken, the first experiment we did for this was to mix two completely divergent cells, K562 and a erythroleukemic line, which represents the mesoderm lineage, and HepG2, a hepatoma line, which represents the endoderm. Basically mix them together, and then 200,000 cells, and mix them together at various ratios, 200,000 cells per reaction, various spike-ins. We're adding in E. coli, we're also adding in spike-ins. In addition to actually the spike-ins being used to monitor experimental specificity, they can also be used to normalize the experiment, because you know exactly how much you put in every reaction. When you do this, you can actually see the data appearing and disappearing. Here's the K562 signature disappearing as you increase the HepG2 percentages by 20%, and the same the other way around. You can see genes that are specific in HepG2 disappearing as you go to K562. You can see it in the IGV tracks. How does it actually look with regards to the how does it look with regards to the R squared values? And you can see is depending on how we do the normalization, RPKM, E. coli normalized, and barcode normalized, we can always actually get a good Pearson, and we can also see the actual changes that we're looking for. This is not Pearson, it's an R squared. So how about in terms of our sensitivity? Are we only capable of doing this with the top 2 or 1% of genes, or we can actually, or peaks, or can we actually go the whole way down? So here we're looking at 3 different peak strengths, and does TSEP, CJES essentially. And the idea is you can see that I can still get a linear line depending on whether I'm looking at one at the very strong peak or in the top 20% or one in the bottom 20% of peaks. I can also do things for looking at peaks disappearing, or the same thing, I can watch peaks appearing on the other side. So in other words, it does have the sensitivity to actually see relatively subtle changes bioinformatically. How about cell mixing is a bit of a cheat. How about in response to drug treatment? So here we've actually done sort of a similar sort of thing, but now we've actually treated the cells with MV411, which is an inhibitor of menin, which is involved in K4 trimethylation. Same sort of thing. We identify a solid menin dose, maximal dose, and then we mix again at percentages, either untreated or treated at various quintiles. We can show the differential occupancy in these two data sets. Genes are going down, genes are going up with regard, peaks are going down and up in intensity based on K4 tri or menin. And of course, we can actually discern the changes again. So cut and run is a very sensitive technology with regards to looking at change. It's not simply whether the peak is there or not. You can actually watch the response of a peak to an external stimulus, be it a signaling event or an inhibitor treatment. Okay, so as I mentioned, the PTMs don't exist that I was mentioning before. They don't exist to recruit antibodies. We know they're in all these various places. So another major drive that's been going on within the company is to actually ask, well, what binds to these PTMs? And these are readers, which I talked about earlier. The old way of doing reader binding is back to the peptides again. And in fact, the company EpiCypher was established on the basis of distributing peptide arrays, something which we haven't done for quite some years. We also used to do peptide binding services, which we hated because the failure rate for a novel target was around 80%. So we were consistently having to give bad news to people, which was never very pleasant. So we have lots of PTM peptides. So we simply asked, okay, is there a better way of using them? Rather than this peptide array glass slide surface, how about another assay? This is the alpha approach from PerkinElmer. Some of you might have heard of it, but it's analogous to chemical FRET. You've got a donor and an acceptor bead. You excite the donor, release a singlet oxygen, causes emission from the acceptor within a given distance, if it's within a given distance. And you can bridge the distance by a protein-protein interaction. In this case, you can have a biotinylated peptide or nucleosome on the donor bead and a reader domain on the acceptor bead. And if the two engage and you do the excitation emission, you can actually measure the interaction and its strength. Okay, so this is a robust technology. It's homogeneous, which is a nice biotech way of saying no wash. No wash means fast, and no wash means very, very sensitive. So what does the data look like? This is BRD4 bromodomain 1. On peptide arrays, we'd always been using this at around 2 micromolar. And when we did that, we got 17 hits. So we had about 300 peptides, and we got 17 peptides that this thing bound to above background, which literally was above background. And this even was one of the best of it. When we dropped the concentration 2,000-fold, and we used it in this new assay, which we were calling EpiCypher, so 1 nanomolar, we got 25 hits, which it encompassed all the ones that we'd seen. We were simply picking up weaker interactions. If we increased the concentration, we got more and more. Now the question was, is this biologically meaningful? And that's always the question. You're getting lots of interactions. Do they actually mean anything inside the cell? So for us to do this, the first thing we wanted to ask was, okay, what is the comparison between peptides and nukes with regards to the data that's yielded? This is a different reader domain, HP1 beta, which is a chromodomain, which is involved in binding to methylated chromatin, canine methylation. It's a mark of heterochromatin. And so it binds, this is a way of actually representing the alpha, the deciphered data. Blue is an unmodified peptide, a modified H3 control peptide, and red is canine trimethyl, because this binds to canine methylation. The further you are to the left, the stronger the binding, and all we're doing is we're increasing the concentration of HP1 beta. You can't simply flood the system with the query, being the protein that you're trying to, because if you do, eventually it will start to bind to the unmodified peptide. So you have to find the sweet spot. So here are the sweet spots over here. We use this to then probe the entire setup. We've got about 300 peptides all on beads. We get 20 hits. They are all focused on H3, on histone H3, ignoring 2A, 2B, and 4. And every one of the hits contains canine methylation, mono, di, or trimethyl. The only time we don't get a hit on canine methylation if we have adjacent serine 10 phospho. This was known in the literature. Canine methylation, mark of heterochromatin. There's the mitotic phospho switch. Serine 10 is phosphorylated mitosis, ejects heterochromatin proteins, which then have to reestablish heterochromatin in the next cell cycle. So this all makes perfect sense. And this is an example of what had been seen in the literature. This is an old peptide pull-down experiment from years ago. And what you do see is the binding to all 3 methyl states of K9, ignoring largely K20 and a significant background on H3K4. You can get a relative idea of the signal noise here, but this is what the signal noise looks like in the alpha setup in the EpiCypher. That is unmodified down here. That is 50 to 100,000 counts. This is undetectable. It's tens, maybe 100 counts. Massive signal over noise. And what you yield with this data, HP1 beta to peptide binding to peptides. Now let's start thinking about the biology. HP1 beta binds to K9 di and tri. It doesn't particularly care about either state. If you drop to a lower methyl state, it binds about 50%. Biologically meaningful, who knows? But if you're a demethylase and you're converting from tri to di, you should have no impact on HP1 function. The only problem is, when you move to nucleosomes, this is not what you see. This is binding to the K9 trimethyl in red, the unmodified in blue, find the concentration, probe the same methyl nucleosomes again, and now what you immediately see is, again, beautiful signal over noise, absolutely no binding to the lower methyl state. It is completely ignoring it. This changes the biology now completely. K9, it is a trimethyl binder. It is not binding to di or mono at all. So this changes the way that we approach investigating HP1 beta function. There is so much data in this going through the lab down the years. This is L3-MBTL1. Very interesting and very strange. It's a 3x MBT domain. It binds to lysine mono and lysine di and does not care about the residue. So here, for example, is it's binding to H4K20 dimethyl versus a control. Find a concentration, probe the array, 90 hits. Every single one of them, H4K20, K9, K4, K27 monodi, ignoring the tri. Very strong data, exactly as the literature would say. Except, think about it, it makes no sense. These are completely different PTM targets. They're found at different locations, they're regulated in completely different ways. Why would you need a reader that can bind to all of this? What possible function could this serve inside cells? And the thing is that actually when you put it onto nucleosomes, it binds to none of them. In other words, the biology is now shifted again. This is not a K20 reader. It is not a K4, K9. It's probably binding to something, but it's definitely binding to these. Its relationship with chromatin is not via the targets that it's currently believed to bind. So the system is completely modifiable, titratable. We can do 2D titrations of the protein concentration versus salt concentration. Here I'm simply doing 4 different queries: a bromo domain for BRD4, Yates domain, which is another acyl reader, HP1 beta, chroma domain binding methyl, lysine methyl, K36 binder NSD2. We'll talk about this in a moment. It's a PWWP domain. And simply we've normalized in each case to the strongest signal. Sorry.
So was that previous slide indicating that the PTMs and the peptides are just getting hidden in the nucleosomes, so that L3-MBT can't bind them anymore?
Precise. That's the assumption, yeah, that in a peptide context, in fact, we've got tons of data now suggesting this. In the peptide context, the target is there. It's just visible. In a nucleosome context, it's not. And therefore, L3-MBTL1 can't engage because they're protected by the nucleosome surface from its engagement. That's the current thinking on this.
Okay.
So there's presumably a threshold event. Yeah. Nucleosome engagement is regulatory. Essentially, the fact that the target is plastered onto the surface is itself regulatory to downstream events. And in fact, there are different enzymes that can drive the peptide off the surface to make it available. So that's one of the things we now believe acetylation is actually doing, in addition to being, and we'll come to that in a moment. So what you can see in each case, we're looking at the unmodified control nuke versus the ostensible target. We're getting a beautiful signal that we can see signal over noise. In this case, in the PWWP domain, what you can see is we can never actually get this state. And this is because PWWP domains have actually 2 points of contact. And that's another thing to keep in mind. PTMs never work on their own. There are multiple points of contact, can be with other post-translational modifications, this being the histone code, but there's also other nucleosome surfaces. The common ones for non-specific engagement or differential engagement are the acidic patch and the DNA. PWWP domains actually bind to K36 methylation, but they also bind to DNA. And what you're seeing here is the fact that it's DNA engaging, and you can't actually get the PTM engagement above this threshold. And the way you actually do it in the assay is you throw in a blocking reagent, just like you would do when you were doing a southern blot, which probably most of you don't even remember what they were. And so a southern blot is salmon sperm DNA. And that's exactly what we've done here. We're simply adding into a different PWWP domain. We're adding in salmon sperm DNA, and what we're actually watching is the signal over noise, the window appearing as we actually add in the blocking agent. Whereas this has no effect on something like AF9 Yates, which doesn't have a DNA binding mechanism. We've actually used this to interrogate the PWWP domains. And so some years ago with Dave Ellis, Jacek Majewski, and Chalu, we identified the PTM binding preference of DNMT3A involved in DNA methylation. And we showed that it was actually binding to K36 di and K36 tri, which explained why DNMT3A localization overlapped with these PTM targets. And it also explained the reason that it's in nature is because it ties together 2 complete arms of epigenetic regulation, DNA methylation and histone modifications. You modify K36 via NSD12, This gives you the K36 target, which is then bound by DNMT3A. Essentially, one DNA methylation in this case is downstream of the PTMs. This also explains when you have mutations or hypomorphisms in NSD1 leading to diseases like Soda syndrome, why you've got a similar phenotypic pattern when you see mutations or hypomorphs of DNMT3A, Tappan-Brown-Raman syndrome. Okay, so one thing I've mentioned briefly in passing is the fact that PTMs never travel on their own, they never work on their own. You've heard about you've heard about the histone code, etc. If you ever look at a PTM binding engagement of a reader, it is milli or micromolar. That is really important. It's presumably because to force it to function cooperatively in vivo. And so the idea here is that we believe that different PTMs are traveling together and they have to be engaged simultaneously to engage a physiological or cellular mechanism. An example of this would be BPTF, which at its C terminus contains a PhD, which binds to, is a methyl binder, and a bromo, which is an acetyl binder. And so here we have, from the literature, we know that, this is BPTF in its larger context, and it's also part of a larger complex called NERF. But what we have shown over the years is that the PhD binds to K4 methylation, and the bromo binds to H3 and H4 acetylated tails. And so therefore, this has been shown by us and other colleagues and also been published. Therefore, you would assume that if you have a peptide containing K4 tri, you'd get PhD engagement. A peptide containing this acetylation, you'd get bromo engagement. And you should get synergistic engagement, or at least additive, if you have what we call the trimethyl triacetyl, because you're now engaging the two of them together. And when you look at the peptide data, sadly, this is not what you see. Here is the binding to the unmodified in blue. The bromo acetyl engagement is in purple, the methyl PhD engagement is in red, and then in green, you're not seeing any indication of additive or synergy. These two things appear to be binding independently of each other. The histone code in this case does not appear to be working, which makes no sense. However, when you move to nucleosomes, again, everything changes. There is the binding to the unmodified, the PhD in purple, the PhD in red, the bromo in purple, and this is a log increase in binding in green. On the nucleosome, it is showing synergistic engagement. So we've done all this work with Catherine Musselman, and so we were going to do then, when we were putting this together for publication, you can imagine what we were doing. We had antibodies to BPTF, we had antibodies to the methyl, antibodies to the acetyl. We would then show co-localization in the population in a cut and run experiment, and then we would begin to argue with the reviewers about, well, it's a population. You're showing they're in the same place and you're showing me stats, but how are you proving that it's in the same place in the same cell? This is a log increase in binding. Question, is that experimentally meaningful with regards to a genomic mapping experiment? So we can make nucleosomes. This comes back to, we can actually make them and we can spike them into an experiment and we can look at their performance inside the experimental setup. So we created nucleosomes containing the targets of BPTF. And so we have one K4 tri, tetraacetyl, so this is for, of course, the PhD, for the bromo, and this is trimethyl triacetyl, so this is the dual engagement, the tri-tri. And we took the same biological tool we had a moment ago, being the GST PhD bromo, and we simply added an antibody to GST. This is reader cut and run. You're taking the same experiment you would have used for a pull-down experiment, you're simply throwing it in and asking, how does it do inside the genomic cut and run experiment? And when you do that, and the first thing you of course do is you look at the recovery of the spike in standards, because they're barcoded and they can tell you if there's dual engagement. And so here we're looking at IgG, you're seeing no preferential barcode release, indicating exactly this is the background. An antibody to endogenous BPTF, of course that doesn't work because that's basically going after the stuff that's inside the cell engaged to its target. This is GST BPTF. You can see that it is not releasing the unmodified, it is not releasing the tetracetyl, it's doing weak release of the K4 tri. What it absolutely adores is the nucleosome that has both PTMs in the same place at the same time. This is dual engagement, stabilized binding, cutting and release into the supernatant. So how about, these are the standards, how about inside the cells, do you actually see a peak structure? And you do. This is K4 tri, this is K18 acetyl, one of the representative acetylations from the triacetyl panel. This is endogenous BPTF, and this is exogenous to stuff we added in. And you can see they are strikingly similar. It's not simply noise, you actually get a beautiful peak structure. What you also see is the vast majority of the data set is you need methyl and acetyl at the same time to recruit endogenous and exogenous. If you have abundant acetylation, it is not sufficient to recruit either target. So in other words, you do need dual engagement. The vast majority of the data looks like this. We are extremely interested in data sets that look, the very small number that actually look like this, where you have abundant methyl, abundant acetyl, and you are recruiting the exogenous, but you are not recruiting the endogenous. Why is this happening? We believe this is actually the next step in the histone code. It is not all exclusively additive. Remember, we are simply looking at this region here. There are loads of other domains spread across these proteins. It's possible that some of these are seeing a negative signal. And in fact, it's looking at this location, the endogenous, and going, okay, I've got the two things that the PhD Bromo likes, but there's something else in there that I do not like, and I will not go there. This is the next stage in the histone code evolution. So we have about 5 or 10 minutes left, perhaps. So we've got enough time to do a bit of R&D, and I'm going to show you some of the other shiny stuff. And I'm going to look at very briefly over 2 stories. One of them is DNA methyl, and it's actually multi-omics. And so up to now, I've been talking about post-translational modifications. I've talked in passing about DNA methylation, that work we did on DNMT3A. But we've been working a lot in the last few years with NEB, New England Biolabs, and we've been trying to tether some of our technologies together. And the first one we did, some of you who work on DNA methylation are probably familiar, you've heard of bisulfite, which is sodium bisulfite treatment and then doing whole genome analysis. And the idea there is that you add it to cells. If cytosine is unmodified, it gets converted to a uracil, which is then read as a T. If cytosine is methylated by either 5-methylcytosine or 5-hydroxymethylcytosine, it is protected from the bisulfite, and therefore it is read as a C in subsequent analysis. And in this way, you can actually map 5-MC, 5-HMC as a pool. NEB developed a technology called enzymatic methyl sequencing, which does the same thing, except instead of using bisulfite, which is extremely damaging to DNA and fragments it, and makes life really complicated for subsequent analysis, you have a 3-enzyme cocktail which basically achieves the same end. You add them in and it converts. The only thing is that by leaving one of these things out, you can actually also distinguish between 5-MC and 5-HMC, which is really important when you're trying to map the two of them independently. There is a technology known as ChIP-seq bisulfite, and it's exactly as you'd imagine. You enrich for a target with ChIP, and then you take the DNA and you try and map the DNA methylation substatus of it by doing the bisulfite sequencing. The problem is, it's 2 crappy technologies linked together. ChIP is terrible, bisulfite's also terrible. So basically what you end up with is even worse. It is, however, true multi-omics. You're seeing, I enriched for A, and now I'm looking for the methylation status. You then have to determine if the relationship between the two is direct or indirect, but at least you have true multi-omic insight. Cut and run is a great technology, and EM is also a great technology. So essentially we've tethered these 2 things together into a workflow. And the one beautiful thing is we can control both steps. NEB has taken exactly the same approach as we have. We control the enrichment for the cut and run with the spike hits. So in other words, we can tell if the experiment has worked or not. And I've shown you we can do that for any target we want. NEB has taken essentially the same thing. By sulfite, you can tell how effective the conversion has been. So if the conversion is ineffective, you're now reading it as methylated or non when it wasn't actually that way. But they actually spike in DNAs, which either contain 5-methylcytosine, 5-hydroxymethylcytosine, or unmodified. And in that way, they can tell exactly how far the experiment has gone and if you should actually be reading the data. When you take the 2 of them, you can actually look at differential feature association. So here, for example, I'm looking at, this is the global abundance in K562 cells of 5-methylcytosine. If you look at the total levels. And what you now see is that if I enrich with a given target, the numbers shift. That is the IgG, agreeing with the global abundance level. That is K4 tri, and what you can see, it is dramatically underrepresented. In other words, DNA methylation is not found in most promoters. It is specifically kept away, which makes sense about what we know about DNA methylation. K9, you can see the association. K36 methylation, you will not be surprised to see is associated with DNA methylation, because I just showed you a while ago that K36 methylation di and tri is involved in the recruitment of a DNA methyltransferase, so it makes perfect sense. And BRD4 CTCF. The one joy about this is it takes the strong advantages of both tech. I told you you only needed, for example, 5 to 8 million reads to do this. You only need the same amount of data to actually do this multi-omics approach. Here are various tracks. They look very similar to what you've seen for cut and run, or you would expect to see in an IGV browser, except what you're actually looking at is the methylation status of the DNA. Unmethylated in blue, methylated in red. And therefore, no great surprise to see that K36 track heavily enriched in methylation, because this mark is involved in the recruitment of a methyltransferase. On the flip side, K4 trimethyl, these promoters are active and spared from methylation. This is downsampling. How low can you go? How much data do you actually need to generate to look at a DNA methyl signature? That is 100 million reads. You guys here have a NextSeq 2000. That will generate anywhere from 400 million to 1 billion reads, depending on the cartridge you use. 100 million reads would be a lot of them. You could multiplex only 4 to 10 in this experiment, but however, we can actually go the whole way down to 5 million. Now you're multiplexing up to 200 individual reactions and looking at the DNA methyl status in this experiment. This is true multiomics, and it is true cost control. In other words, you can do massive amounts of target enrichment and then look for the relationship between factor A and factor B. That includes mutation status, etc., etc. We again are trying to control it. Here, coming back to, I showed you a few moments ago, I was using BPTF for reader cut and run. You can also do reader TempSeq. TempSeq, by the way, is our current catchy internal name for targeted enzymatic methyl sequencing. MECP2 is a transcriptional repressor, but what is known is that it actually is recruited, it's got a methyl binding domain which binds symmetric 5-methylcytosine. In other words, both strands are actually methylated on the CG. And the flip side of that, by the way, is hemimethylation, or only one of them is, which occurs during cell division. So here, for example, we are looking at reader cut and run, or reader TempSeq, using GST MECP2, binding to the DNA methyl standard, be it unmodified, hemimethylated, or symmetrically methylated. What you see is this massive enrichment, or this strong preference for the methylated standard over the hemi. This, by the way, is 135x. That is even better than an antibody would be. And that is an exceptional antibody with showing target preference for K4 trimethyl versus unmodified. These things have exceptional capabilities. Antibodies are great, but you can actually repurpose the biological things themselves to actually do genomics assays if you're careful. And of course, when you look at the data, what you can actually see is here's IgG. Basically, here's a region that's methylated, here's a region that's not, and what you see is GST and MECP2 is, of course, enriching inside the region that's methylated, and when you look at the DNA underneath it, it is also methylated. The final thing I'll call attention to for the last few minutes is NICE. This is open chromatin mapping. The standard technology, just like the standard technology used for DNA methylation, that methylation mapping is bisulfite, a tech that's been around a long time and is pretty it's got its strengths and minuses. The standard approach that's used for open chromatin mapping is ATAC. Everybody's heard of it, assay for transposon. So essentially it's TN5. And so what the TN5 is doing is you add it into an experiment, it's got its DNA cargo on board, it goes looking for regions that are open. When it finds them, it tagments, it drops its cargo at that location, and then basically you go looking in the genome sequence for the actual cargo. You can in fact PCR between the dropped adapters, that then creates a library which is then a sequence. And by mapping that back onto the genome, you can identify locations that were open. It has been around for quite a long time. It has strengths and weaknesses. One of the weaknesses that it has is that in the early iterations, there was tons of mitochondrial reads. Most of that has now been sidestepped with improved technologies. But the one thing is it's very, very poor at cross-linked material. Cross-linked material, there are reasons you might want it. You might want it, for example, if you're doing drug inhibitor treatment, because you capture the situation, particularly if you're going to ship the material from the place it was created to the place it was analyzed. But the cross-linked material that we all want access to, if we're doing drug development, etc., is FFPE. This is banked material coming out of tissue biopsies. If that material was accessible to any type of genomic approach, there are tens of years, or like millions upon billions of samples that are available for analysis. So we have been working with NEB on an alternate technology where, in addition to ATAC, called nicking, or nickase, nice sequence, and nicking enzyme-assisted sequencing. And it's a nickase enzyme that you add in. It does the same thing. It looks for regions of open chromatin and preferentially cuts there. It's basically a simple enough experiment. There are multiple generations of it. I think we're currently on the 3rd or 4th generation, which is known as one-pot. And the reason we like this one is you add in the nicking enzyme, it finds the open region, But then you actually try and protect it and label this region. So you add in addition to doing the nicking, you then do a fill-in experiment with polymerase, which fills it in with biotinylated CTP and also adds in a methyl CPT, which protects it from subsequent nicking. So at the same time as you're doing the nicking, you're also doing the labeling and the protecting step. You then basically reverse the crosslinks inside the same tube with thermolabile PK, and then you basically add in the nicking enzyme again, and now it cuts the rest of the genome to ribbons. That's important because you're doing it all inside the same tube. A previous iteration, for example, required you to do sonication. That's fine if you're doing one at a time, but if you're trying to do 10, 20, 30, 40 samples, you do not want to be moving from one place to another. You want to be able to do it all in the same tube, hence the one pot unionize. So essentially, the second round of nicking, the label regions are protected because the CTP is there and the biotin is there. And then you simply enrich for these on streptavidin beads, you prepare libraries and you sequence them. So what does the data look like? It's pretty spectacular. In fact, here we're doing, looking at 5,000 cells, the peak strength at transcription starts like 5,000 cells all the way down to 25. Beautiful data the whole way down. Same thing at enhancers start and end sites, you're getting beautiful data. You can also look and ask, well, as I drop the cell sequence, as I drop the input material in that levels, do I actually lose some data from specific locations which are presumably more weakly open or more weakly cut? You don't. You're getting equivalent representation as you drop the data down to fewer cells. You can also, of course, everybody wants to see the peak structures. You can, of course, look at the tracks, and you're seeing beautiful peaks at 5,000 cells all the way down to 25. You're getting equivalent data. You can go back and say, well, okay, this thing works on FFPE material. In fact, the standard here is 4% formaldehyde. How about ATAC? Does that work on cross-linked material? In fact, there has been a study done some years ago showing, yes, ATAC does work on cross-linked material. Yes, it does, but it's extremely weak in comparison. Here is one pot union-seq at 25 cells. That's the data set, the strength of the peaks at those locations. That's with 500. That in comparison is the ATAC-C, which is what the tech is known, at 50,000 cells. So you can see as you are yielding data, but is much weaker data. And of course, everybody wants to see what the peaks look like. And there is, of course, a dramatic difference in those between the signal noise and what you would call as an open or closed location. But the big thing is, how about FFP? Now, this is where the strength, our current focus is lying within EpiCypher. All FFP material is not the same. You hear about it, if it's been collected, it's relatively fresh, it's been stored appropriately, that's probably fine, or at least that's the assumption, that it's been stored appropriately and it's about a year or two old. Older than that, you get massive fragmentation. So something that's sitting inside a filing cabinet that's been there since 1975, this is not going to be suitable for this because that material is actually lashed to ribbons. But here we've simply taken FFP material from a human liver, there's 5 micron sections, 2 replicates, R1 and R2. They're deparaffинized, rehydrated, and then put into the experiment. And then basically you just do the genomic analysis. You look at the comparison of the enriched features, you look at the chase maps of this is basically the peak strength at transcription start sites and enhancer elements. Everything looks fantastic. The only thing that we're not too thrilled about is the replications. And we're not sure if this is because replicate differences or because the bioinformatics are entirely set up for it. So this is a major focus. So internally, FFP shows massive promise, but until it is stronger than this degree of overlap, we are not classing it as fully fit for purpose or roll out into the community. Hence why it's still inside R&D. And of course, you can look at the peaks. You are seeing these are beautiful real peaks. And then the final thing, of course, would be, can NICE be used for drug development testing? This is what most of us are doing. You've all heard about the relationship, perhaps. Members of the SMARCA complexes are some of the strongest. They're essentially heavily mutated in various cancers. And so therefore there's been a massive drive to develop inhibitors of BRG and BRM, which everybody believes will have great drug potential. For example, local companies like Foghorn were established initially to set up for remodeling inhibitor development. Here we've got cells which are BRG1 mutated, treated with BRM014. This is an inhibitor compound that was developed some years ago, a tool compound by Novartis. And this is a dual inhibitor of SMARCA2 and SMARCA4, which is the other names for BRG and BRM. In this case, we have 8 reactions. Each of them are 100,000 cells in replicates and fixed, and we're looking at the cells that are untreated, DMSO treated, and then BRM014 at 2 different doses, and looking at the replicates. The Pearson's there, about greater than 0.95. In other words, the replicates look great. And of course, then you do differential peak analysis. Now, what you discover is that there's a lot of things that go up, and a lot of peaks that disappear in response to the treatment because the region is closing when you're inhibiting the actual ATPase, which is normally keeping it open. And so you pick a few of these, and in fact, when you look at it, one of the most interesting ones is a biomarker, which is at the KRT80 locus. And you can see here there are 3 locations that are open in untreated cells. And when you actually treat this with BRM014, this region's closed. And the reason this is called a biomarker is it's something that we can use for the development of new inhibitors, because it's been shown that this locus, this functionality is directly reliant on BRM function. Because the mRNA is downregulated on BRNA knockdown by RNA-seq, BRM is directly recruited to the locus, and this was originally done by ChIP-seq, and the open signature is reduced either on inhibition of BRG-BRM or by knockdown of the actual target of the protein itself. So this is what would now be known as a biomarker. So in other words, if you're developing a new inhibitor of these things, you would basically treat with the inhibitor and then ask whether this locus is affected. That tells you if you're on or off target. All right. So you get a general feel for how we're applying the tech. So I would like to thank all of my collaborators, or all of our collaborators at various stages involved in the antibody testing. This is the DNMT3A studies. Colorado is actually work I didn't show you today about ubiquitination DNA damage. We also didn't talk about today, but primarily also our colleagues in New England Biolabs and all of my colleagues in EpiCypher. And I'll take any questions, and I'm around for lunch or subsequent. Thank you all very much.
Let me start with one quick question. You mentioned FFP, tissues and stuff. In your diagram, you always have to fix the cells to the solid support. Is that single cells, or can that be like clumps of cells?
You mean in terms of the slide? Oh yes,
You're
oh
right,
yes.
because in your katana,
No.
you always have things.
Yeah.
Like, can this work for tissues, or do you have to have a single cell suspension?
You don't need a single cell well. So for a cut and run experiment, yes, you need a way of immobilizing it, and you need a way of immobilizing the standards as well. But yes, you need to have access. So generally, we're going after, mononuclear suspensions. But because of course, if you've got clumps, if the antibody can't get in and the enzyme can't get in, then it can't find its target and nothing can get back out again. So you need accessibility. If you had large clumps, it would depend. If, for example, if there were clumps in the system, it would probably be okay. But generally, we're going after mononuclear. Clumps are never a good thing when you're doing these experiments.
But ChIP-seq, you can kind of get away with clumps.
ChIP-seq is, because in a ChIP-seq you've blown everything to pieces. You basically take the material, be it a tissue, and then you blow it to pieces with sonication, with MNAs, with SDS, with etc. That has advantages and disadvantages.
Okay.
Yes, actually, I have 2 questions. I actually first came across you guys a couple of years ago at AACR, and it's really exciting to see how far your science has come since then. I think it was like 2018. I came across some EpiCypher booth at AACR. And so I have 2 questions. One, in your later slides, you started showing, TSSs, enhancers, things like that. But in light of your more recent results, that our old ChIP-seq definitions of these regions may be not so nuanced, to say the least, have you guys attempted to redefine those classical genomic elements?
Yeah, to a degree, internally, but not externally.
Not externally.
So enhancers are classically, enhancers are genomically defined. If you do it by PTM, it's generally by K27 acetyl enrichment and also by K4 mono.
Yeah, and now there's like Chrome HMM. I think
there's other ways.
More, which actually I had to use, which is why I was asking. I'm just kind of curious, if internally you guys obviously.
For example, the K27 acetyl antibodies that the field is so in love with, which is one of the major defining things. We've discovered that the vast majority of those are actually not specific for K27 acetyl. They're hugely influenced by polyacetyl signatures. And if you take another antibody that is truly K27 acetyl and you do them in parallel, you discover that the standard antibody is looking at 2 different types of element. And so we're currently trying to define with collaborators, what is the distinction between them? So not all enhancers are the same, but you can get beyond them. They're not all simply defined by PTM. If they were, I think I'd be very concerned. They're now also defined by the open chromosomes at those locations, and now also further by the presence of various remodeler complexes and TF factories and whatever you want to call them.
Just one more question before we do the lunch in the back. Anybody else have one more question? One of you choose.
Yeah, so what about the cell types sensitivity? Because I know some cells are more sensitive to the content. For example, keratinocytes are very prone to infiltration of the antibodies inside the cell. Are there some modifications for the cell type specificity of the reaction?
We're in the middle of actually exploring that at the moment. Up to now, we have a standard protocol that was used for everything. And we've discovered that that's not entirely appropriate. So for example, macrophages are full of phagocytic granules. If you do a standard cut and run experiment with those on intact cells, basically the granules spend their time trying to destroy the entire experiment on you. You get huge noise. And so for that reason, with macrophages, we say you can only do it with nuclei. And in fact, you've got to do a pretty good protocol to remove as much as the cytoplasmic fragments. We've not talked about keratinocytes, so I'd imagine neutrophils would follow a similar macrophage problem. We also have some internal data with ES cells indicating that because they have a more open general chromatin structure, they might require modified protocols as well, but that's something that's being discussed internally. If anybody has any feedback to give us on other cell types and what you've done to modify the protocol, we'd love to take a look. Our services group, our R&D group has their own preferred cell types, and of course, as you can imagine, but our services group does not. And so we've probably taken in close to 70 or 80 different cell types. The majority of those came through the ImGen project. It was, I think, up to 70 or 80 different 10,000 sorted cells of various types. And the majority of that data was pretty straightforward. We didn't see any issues. But the cell numbers there were only 10,000. And then the macrophage problem, for example, wouldn't have reared its ugly head because there just wasn't enough material to cause the assay to go down.
Okay, thank you, Dr. Cho, for that